Hello aspirants, welcome back to our channel FoodTech Online. Hope you all are doing good. So today will be our sixth class for the most important questions for the entrance examination, that is for PG entrance examination in food technology subject. So let us proceed. So question number 1, group of salt-loving microbes are called. So options are mesophiles, halophiles, saccharophiles or heterophiles. So over here my correct option will be option number B, that is halophiles. So halophiles, these are the group of salt-loving microbes. Okay, whereas what is mesophiles? So mesophiles, actually these are the microbes that thrive to stay in the moderate temperature, whereas saccharophiles are the microbes which thrive to stay in the cold temperature. And heterophiles, so heterophiles, it is not a recognized term in microbiology. So over here my correct option will be option number B, that is halophiles. Next, question number 2. Fish hydrolysis are rich in. So options are proteins, lipids, vitamins and minerals. So over here my correct option will be A, that is proteins. That is fish hydrolysis, these are rich in proteins. Actually this fish hydrolysis, they contain small amounts of lipids, vitamins and minerals, but the main component is the protein. Actually they are valued for their high protein content, which can provide the essential amino acids, and it can even support the biological functions in the organisms. So over here my correct option will be A, that is proteins. Question number 3. Endospores are produced only by, so options are gram-positive bacteria, gram-negative bacteria, both of these or none of these. So over here my correct option will be A, that is gram-positive bacteria. So endospores are produced only by gram-positive bacteria. These are primarily produced by gram-positive bacteria only as a survival strategy in response to the unfavorable environmental conditions, whereas these gram-negative bacteria, they do not produce endospores as a means for their survival. So over here my correct option will be A, that is gram-positive bacteria. Question number 4. Production of bacteriocin in food, it is considered as an, so options are implicit factor, intrinsic factors, processing factors or explicit factors. So over here my correct option will be B, that is intrinsic factor. So production of bacteriocin in food, it is considered as an intrinsic factor. Why? Because it is inherent to the food itself, and it directly affects the food's stability and its safety. That's why it is an intrinsic factor. So over here my correct option is B. Next, question number 5. The storage polysaccharide is, so options are glucose, glycogen, glucagon or glycated HB. So over here my correct option will be option number B, that is glycogen. So we know that glucose, it is a simple sugar and the primary energy source for the organisms. Glycogen, it is a complex polysaccharide that can serve as a storage form in the animals. And glucagon, it is a hormone which is involved in the regulation of the blood sugar level. And what is glycolated hemoglobin? So it is actually a marker which is used to assess the long-term glycemic control in the diabetics. So over here my correct option is B, that is glycogen. Okay. Next, question number 6. Cofactor of an enzyme blocks the enzyme activity, assists enzymes in catalysis, or enhances the enzyme activity or increases the activation energy of the enzymatic reaction. So over here my correct option will be actually this cofactor of an enzyme, it assists enzymes in the catalysis as well as it enhances enzyme activity. So over here both of them are correct, but the most appropriate one is that it enhances the enzyme activity. So they do not block the enzyme activity or they increases the enzyme activation energy of the enzymatic reactions. Rather what they do, they contribute in increasing the efficiency of the enzymatic reactions. How? By aiding in the catalysis. So over here my correct or the most appropriate correct answer will be C, that is it enhances the enzyme activity. So next, question number 7. The principal buffering compound in milk are, okay. So the principal buffering compound in milk are fat, lactose, salts and proteins or water. So we know that milk actually it contains fat, then lactose, that is milk sugar, and water. This fat and lactose actually they do not primarily function as a buffering compound, and water we know actually it contributes to maintain the volume or the composition of the milk, but it do not directly participates in the buffering of the pH, whereas the salts and proteins they are the principal buffering compounds of the milk. So over here my correct option is C, that is salts and proteins. Next, question number 8. So casein in fresh raw milk, it is present in the form of, so options are micellar, globular, solutions or insolubles. So this casein in fresh raw milk, it is present in the form of micellars. So over here my correct option will be A, that is micellars. Actually this micellar form of casein is crucial for various properties of the milk, like it includes the textures or the nutritional quality or processings or other characteristics. So over here my correct option is A, that is micellars. Next, question number 9. The enzyme which is used to detect hydrogen peroxide in milk. So options are lipase, proteinase, catalase or lactoperoxidase. So over here my correct option will be option number C, that is catalase. So catalase, it is an enzyme that can catalyzes the decomposition of hydrogen peroxide, that is H2O2, into water, that is H2O, and oxygen, that is O2. So, whereas this lipase and proteinase, these are the enzymes that catalyzes the hydrolysis of lipids and the proteins. Whereas lactoperoxidase, these actually it is an enzyme that is found in the milk and it plays a role in antimicrobial defense systems, but it is not typically used in the detection of this hydrogen peroxides. So over here my correct option will be C, that is catalase. Next. So question number 10, quantitative PCR, that is qPCR, it is also known as. So options are multiplex PCR, nested PCR, real-time PCR or touchdown PCR. This is a good question. I'll give a star to this. So over here my correct option is C, that is real-time PCR. So this, yes, we'll define all these options. That is, what is multiplex PCR? Actually, it amplifies multiple DNA reactions simultaneously but in a single reaction. Okay. Whereas nested PCR, so nested PCR, it involves two successive PCR reactions where the product of first PCR, it serves as a template to the second PCR, which even provides the increasing specificity or the sensitivity to the reactions. What is real-time PCR? That is qPCR. So qPCR actually it amplifies and quantifies the DNA in a real time during the reaction, which allows the detection and measurement of the specific DNA sequences in a sample. So it has various applications. So what are the applications? It is in the gene expression analysis, pathogen detections or genetic testings. Whereas this touchdown PCR, so touchdown PCR actually it decreases the annealing temperature in successive cycles to increase the specificity, and even it decreases the or reduces the non-specific amplifications. Okay, so over here my correct option is C, that is real-time PCR. Next, question number 11. Lipid oxidation during early stage is due to, so options are free radicals, browning, lipoxygenase or processings. So over here my correct option is A, that is free radicals. So this lipid oxidation actually it is initiated by the presence of free radicals, which are highly reactive molecules, and it contains the unpaired electrons. Okay, so whereas this lipoxygenase, so lipoxygenase we know that it can catalyze the lipid oxidation. It actually plays a typical role in the later stage of the oxidation. Whereas what is browning? So browning we know it is a reaction that takes place in the carbohydrates. And the processing, so processing it can influence the lipid oxidation, but it is not a primary cause during the early stage. Rather this processing it can introduce the conditions that promote the free radical formation or lipid oxidation in it its later stages. So over here the early stage is due to the free radicals. So option number A will be my correct option. Next question number 12. So high pressure liquid chromatography, that is HPLC. So it is a dash technique, that is separation, novel, vacuum creating, or a high-tech technique. So over here my correct option will be option number A, that is separation technique. So we know that HPLC, it is indeed a high-tech technique, but its primary function is to separate the components in a mixture rather than being solely defined as its in its technological sophistications. So over here correct option will be A, that is it is a separation technique. Next question number 13. Damage caused to the proteins by irradiations of food. It includes formation of nitrogen oxides, formation of actomycin complex form actins and mycins, and breakage of peptide bonds or formation of lipoproteins. So over here my correct option is A, that is breakage of peptide bonds. Sorry, this is option number C and this is option number D. So correct option is A, C, that is breakage of peptide bonds. So this irradiation of food, it can cause damage to proteins by breaking the peptide bonds that links to the amino acids together in a protein structures. So this high energies like the gamma rays or the X-rays or electron beams, it can include these chemical reactions and can disrupt these proteins structures, the molecular structures of these proteins. So option number C is my correct option. Next question number 14. Eicosapentaenoic acid, that is EPA, is saturated fatty acids, omega-6 fatty acids, omega-3 fatty acids, or essential amino acid. Okay. So the correct option over here is C, that is omega-3 fatty acid. So this a eicosapentaenoic acid, that is EPA, it is a long chain polysaccharide, polyunsaturated fatty acid which belongs to omega-3 family. So option number C is my correct option. Next question number 15. Fats, lipids are transported in blood in combination with proteins in the form of, so options are lipoproteins, cholesterols, phytosterols, or lecithins. So over here my correct option is A, that is lipoproteins. So these lipoproteins, these are the complex particles which are composed of lipids such as the cholesterols or the triglycerides and the proteins which is the lipoproteins. So whereas this cholesterols, phytosterols and lecithins, these are the components of this lipoproteins. So over here my correct option will be option number A, that is lipoproteins. Question number 16. Nutritional quality of meat includes high level of saturated fatty acids, low level of cholesterol, low level of sodium, or poor supply of vitamins. So the negative or nutritional quality of meat, it includes the high level of saturated fatty acid, that is option number A will be my correct option. Even it also supplies poor vitamin contents also. Okay. Next question number 17. The conversion factor for nitrogen to muscle protein is, so options are 5.5, 5.8, 6.25, or 6.75. So the correct answer is C, that is 6.25. So for example, we can say that if a food sample, it contains 1 gram of nitrogen per 100 gram of the sample, so it is assumed that 6.25 gram of protein per 100 gram of sample. So this conversion factor, actually it is widely used in the nutritional science and food analysis for this protein calculations. So correct option is 6.25, option number C. So question number 18. Exposure of meat to high temperature in canning, it imparts dash in meat. So options are rancid flavor, peanut flavor, sulfhydryl flavor, or meaty flavor. So over here my correct option will be option number D, that is meaty flavor. So we know that when a meat is exposed to high temperature during canning, it undergoes other various chemical reactions also, like canning or the Maillard browning reaction, caramelization reactions, but it imparts a meaty flavor also. Okay, so option number D is my correct option. So question number 19. So lecithin is most commonly a glycolipid, sulfolipid, phospholipid, or an aminolipid. So it is a phospholipid. So lecithin is most commonly a phospholipid. And another name for lecithin is Phosphatidylcholine. Okay. Whereas what is glycolipids? So glycolipids, actually these are lipids with carbohydrates attached to them. Sulfolipids are the lipids which containing a sulfur group attached to them, and aminolipids are the lipids in which amino group is attached. So over here my correct option is C, that is phospholipids. Next question number 20. So tetrahydrofolate is a coenzyme of, so options are pyruvate dehydrogenase, acetyl coenzyme carboxylase, thymidylfolate, thymidylate synthase, and cytochrome oxidase. So over here my correct option will be option number C, that is thymidylate synthase. So tetrahydrofolate is a coenzyme of thymidylate synthase. Next question number 21. The intake of PUFA should be of dash energy intake. So options are 0 to 5%, 5 to 10%, 8 to 10%, or less than 20%. So over here it should be 5 to 10%. Sorry, it should be 5 to 10%. Okay. So the intake of this PUFA, that is polyunsaturated fatty acid, it should be around 5 to 10% of the total energy intake. Next question number 22. Dash is the only selenoprotein enzyme which is well studied for the biological role of selenium. So options are xanthine oxidase, aldehyde oxidase, sulfite oxidase, or glutathione peroxidase. So correct option is D, that is glutathione peroxidase. So this glutathione peroxidase is the only selenoprotein enzyme which is well studied for the biological role of selenium. So this glutathione peroxidase, actually it is an antioxidant enzyme that helps to protect the cell from the oxidative damages by catalyzing the reduction of hydrogen peroxides. Okay, so correct option is D, that is glutathione peroxidase. Next question number 23. For the laminar flow, the Reynolds number should be less than 2100, 3100, 5100, or 4100. So what exactly this Reynolds number do? So this Reynolds number actually provides information about the flow regime, that is the laminar, transitional, or the turbulent flow. So for the laminar flow, the Reynolds number, it is typically less than 2100. And for transitional flow, the Reynolds number, it should be typically between 2100 to 4100. 4000. And for turbulent flow, the Reynolds number, it should be greater than the 4000. So over here the correct option, that is the laminar flow, the Reynolds number, it should be less than 2100. That is option number A will be my correct option. Okay, next question number 24. For the flow of fluid, we need difference in moisture content, force, concentration, or pressure differences. So the correct option over here will be C, that is concentration. So this fluid flow occurs due to the pressure difference or the gradients between the 2 points in a fluid flow. So correct option is C, that is concentration. Next, question number 25. Hops are used in manufacture of brandy, whiskey, wine, or beer. So over here you have to comment me down the correct option for this question, and let us discuss this question once. So hops are the key ingredients in the brewing industries. Actually these are the flowers or the cones of the hops plant, that is Humulus lupulus. Okay, and these are primarily used for the flavoring stability in the dash production. Okay, so comment me down the correct option for this question. And yes, if you have not installed our app, you can install our app, that is FoodTech Online, and you can practice more such questions over here, and the link is in the description box for you all. Okay, so with this we have completed few important entrance examination, that is food technology subject question answers. And if you like this video, you can share with your friends, subscribe for more such videos. So till then, enjoy learning and thank you.